So the urinary system. I'm sure most of you have come across it. Oops, have you had a look at it in anatomy yet? The kidneys? Okay, I think you're doing that soon. But essentially, it consists of two kidneys, so the kidneys are paired. So you'd have one on the left and the right. This is the kidney over here. Each kidney has a pelvis and then a ureter, which leaves from the kidney. Those two ureters will open up dorsally on the bladder. Now, there's only one bladder, so that's an unpaired structure. And once the ureters empty into the bladder, the urine will leave the bladder via the urethra. Okay, so a way that I always used to remember it is a ureter is two. So there's two ureters, whereas a urethra, where you don't hear that hard T sound, there's only one. Because people always get mixed up between the ureter and the urethra. Okay, so two ureters, one urethra. So this is what the kidney looks like, or the urinary system looks like macroscopically. If you cut it into half, longitudinally, this is what you're going to be looking at. Now this is obviously a sketch, or it's a schematic representation. But essentially you have these triangular structures, which is called a renal lobe. And a whole lot of these packed together, which then empty into the pelvis, is what forms the kidney. So these renal lobes are fused in different animals. Now the only animal that we deal with that doesn't have fused renal lobes would be the bovine. Okay, so this is what a bovine kidney looks like. I don't know how many of you eat kidneys. Never eaten them? No. They're really nice. No. Okay, most often you would be eating sheep kidneys or porcine kidneys. But they do taste very nice. So here you have the bovine. So if you were to eat a bovine kidney, you'd see this in the shop or at the butcher. This is what a cat kidney looks like. The cat has a lot of fat in the capsule, so it has this very yellow color. Here's a canine kidney and an equine kidney. Okay, the sheep kidney looks very similar to the canine. Yes?
The cat kidneys also have a few lobes, not joined.
No, they are fused as well.
But it also has those ridges.
Yes, these ridges that you see here are the blood vessels.
Oh,
Okay, so there's a lot of fat in the capsule, and these are just quite prominent blood vessels that you see there. Okay, so these kidneys look smooth because all the renal lobes have fused, where here you still get these lobes. Okay, cat kidneys, very interesting. Have any of you tried to palpate kidneys? Okay, I think that's something that you will learn. It's only really possible to palpate kidneys in the small animals, so the dog and the cat. If you're palpating a horse kidney, you'd be doing it through the rectum or also bovine, and you can't palpate a sheep kidney unless you've got an arm that thin. Okay, you can't really do a rectal on a sheep. Okay, so you can palpate the kidneys in the dogs and the cats, and that of course is dependent on how fat they are. So if the animal is very fat, you won't be able to feel the kidney. The cats have what's known as floating kidneys. So the kidneys are fairly loosely attached, and they can be anywhere along a specific area in the dorsal abdomen. Okay, so it's normally easier to actually palpate the cat kidneys because cats are smaller and thinner. If you have a fat cat, very difficult. And if you have a cat that's using all 20 of its claws, very difficult as well. Okay, so the functions of the kidney. I alluded to the one function earlier about maintaining the blood pressure. So it's the chief excretory organ in the body. And then it helps to maintain the constant composition of the blood. So why is it important for you as veterinary nurses to have an understanding of how the kidney works? Because one of the things that you're going to be doing a lot as a veterinary nurse is setting up intravenous infusions. Okay, so what are you doing? The very word intravenous means that you're putting something into the venous system. So that's the blood. So what does the kidneys do? They help to maintain the constant composition of the blood. So if you're putting up an IV drip, then you're going to be affecting the kidneys. If the kidneys are not functioning properly, they're not going to maintain that constant composition of the blood. Okay, so you will be confronted with animals that have renal failure. So in other words, their kidneys aren't working. And depending on what type of renal failure they have and how bad it is, you're going to have to adjust what you do with that drip. If the kidneys are also not working correctly and they've failed, there's certain drugs which you will not use as well because the kidneys are unable to excrete it. So it's not just fluid that they're excreting, it would also be breakdown products from certain drugs. Okay, so the kidneys, you'll see also there's a number of drugs which you give to dogs that if their kidneys are not working correctly, you cannot give them the full dose or you just do not use the drug because the kidneys cannot then excrete it. So they're a very important organ and it's very important that you know about them. So now if you think you have this organ, it's sitting there, it's not just cells that are exposed to the environment, it'll have a capsule, as all organs do. So that's the first part that you see. So the kidney is surrounded by a capsule, and that would mostly be connective tissue, and then some smooth muscle as well. Now I've just put this picture in so that you can see what it looks like, but you're not going to be expected to recognize these micrographs. But here you have the capsule, there's some smooth muscle cells, and there'd be a lot of connective tissue in between. And remember I said for the cat, there's a lot of fat in the capsule. So for those of you who go to pathology and you do postmortems on a cat and you pull out this very yellow looking kidney, don't run around saying, oh, I know why the cat died. That is normal. The kidney should look like that. So you should recognize this picture now. So what we've done is we've taken this kidney, it's a canine kidney, and we've cut it in half longitudinally. And you can see that this tissue here is called the parenchyma. So the tissues of organs is called the parenchyma. That just means tissue. And you would be able to see on this picture here that you've got two distinct parts of the parenchyma. The inner part, which is the medulla, and the outer part, which is the cortex. Here it's represented schematically, the inner part, the medulla, outer part, the cortex. And you can see these nice little renal lobes making up the kidney itself. Now, do those terms look familiar to you, the cortex and the medulla? You should have come across them when you did osteology of the bones. Okay, so the cortex is the outer part, and the medulla is the inner part. And that's exactly the same over here. So the cortex you see on the outside, medulla inside. So if we take a look at the medulla, you can see here that it is composed of all these pyramids. So it looks like one continuous structure, but it's actually composed of these pyramidal structures, which are the base of the renal lobes. And they then open up into the pelvis, where the urine collects and will move through the ureters into the bladder. So this sketch over here is quite important for you to know. Again, this is a schematic, but here you can see the renal lobe, and the base of it forms a pyramid. And this is then the medulla. Now the medulla is divided into different regions. You will see here is an outer zone because it's closer to the cortex, and here you have an inner zone which is closer to the pelvis. The outer zone is divided into two parts. You have your outer stripe and the inner stripe. Okay, so all of this is really doesn't sound like it means much. What it is it's parts of the medulla which are differentiated based on their histological makeup. So we'll get to it just now. There's a whole lot of tubes that form these renal lobes that are found here. And depending on which tube is found in which region, that denotes what part of the renal lobe it is. Okay, so we will touch on that just now. But for now, you must just know outer zone, inner zone, and you have the outer and inner stripe. Outer referring more towards the cortex. Then you also have these structures here called the medullary ray. You can see it macroscopically in some of the kidneys, but essentially it's part of the medulla which radiates into the cortex. So you have these little arms over here. They look like fingers almost. And this is medullary tissue, so it contains part of the pipes that carry the urine, and it contains part of those pipes which you find in the medulla, and it projects into the cortex. So hence the name medullary ray. Then this would be the cortical tissue, and that part of the cortical tissue which is situated between those rays is called the labyrinth. Now what is a labyrinth? Have you heard of that term before? It's like a maze. Okay, so a maze is not a straight line. Okay, if it was a straight line, there would be no challenge to it. You can't get lost on a straight line. Okay, if you do, again, you have a problem. So, labyrinth refers to the fact that there's a lot of tubes in here which are convoluted. So, this part of the cortex between the medullary rays contains a lot of these convoluted tubes, therefore it looks like a labyrinth. Okay, so this is exactly what I've just said on the previous sketch. Your outer zone, inner zone, and then you have your outer stripe and inner stripe. Okay, again, this is not what you're going to have to recognize histologically, but you should know which tubules you're going to find in each of these regions. So this is what it looks like. Okay, a lot of pink. But I think you should be able to make out these tube-like structures over here. And we'll get to the slide a little bit later, but this slide is taken from this region over here. And this is of the outer zone of the medulla, and it's at the junction between the outer stripe and the inner stripe. And you can see over here, it looks like there's a difference in these tissues. The kitten is hungry. Okay, if we have a look at the medullary rays, so these are the part of the medulla which project, they radiate into the cortex. And again, you can see here, this tissue looks different from this tissue. Now remember, the part between the medullary rays is called the labyrinth. So can you see here, we have nice straight lines. So those are like rays, they radiate into the cortex. And here you can see a whole lot of convoluted stuff. So that is the labyrinth. Now the reason why I put these pictures in is just to make it easier for you to remember it. Because if you just look at these words and you try and remember it just word for word, you're going to get completely lost and it'll make no sense. So if you actually try and see why it's called this, and you can have a look at the picture, you will remember the words a lot easier, even though you're not expected to identify these structures. Okay, the labyrinth contains the renal corpuscles. That's this structure over here, and that's the beginning of the tubes which start in the formation and the collection of urine. Okay, so I mentioned the parenchyma earlier. So the parenchyma is the actual tissue that makes up the kidney. So what is that tissue? I mean, what will you find in any organ? You're obviously going to find blood vessels, and you will find nerves. Okay, but what are the actual cells which make that organ that particular organ? So in this instance, the parenchyma is a whole bunch of secretory tubules. So what you see over here is literally millions, well, not in this one slide, but a kidney would consist of, depending on the species of animal, hundreds of thousands or even up to 1 million of these tubules. Each round little circle here is the beginning of the tubule. And those tubules do not communicate with each other. So the actual tissue of the kidney is made up by hundreds of thousands of little tubules, which all then end in the pelvis, and then they drain to the 2 ureters and then into the bladder. So this is the structure, the renal tubule, which makes up the parenchyma. Now you can think of it as one continuous tube, and depending on the type of cells that it has and where it's located, it has a different name. Okay, so if you can think of one piece of string, and you've colored that piece of string different colors, each little section will have a name. But it's all one piece. You'll see 2 terms over here. Here you see a nephron, and there you see a renal tubule. So it's important for you to know what is found in a nephron and what is found in a renal tubule. Those are 2 different structures because everything in the nephron has a different embryological origin to everything which is in the renal tubule. So up until here, these tubules all have the same embryological origin, whereas these tubules here have a different embryological origin. Therefore, they're not called part of the nephron. Okay, so this is a very common question which will get asked, is which components form the nephron and which components form the renal tubule. And you'll see a nephron is part of the renal tubule. These ducts of Bellini are the last bit that you get before it exits the parenchyma itself, and that goes into the pelvis of the kidney. Okay, so you should recognize the structure by now. This is one of those renal lobules. So this part over here, over here would represent the cortex, whereas this part down here would be the renal pyramid, which is all medulla. And that bit projecting into the cortex would be the medullary rays. Okay, now I realize that all the things look very small. This is just to give you an overview, and we will zoom up on each section as we go. But essentially, your renal tubule or nephron would start over here, and you can follow that green, and then it goes down the medullary ray, and then it turns on itself in the inner zone, and it goes all the way back up through the medullary ray, back into the labyrinth, passes the same renal corpuscle, then it goes back into the medullary ray, and it joins the collecting tubules, represented here by yellow, and collecting ducts, and then it will exit into the pelvis of the kidney. Okay, and there will be literally hundreds of thousands of these little tubules within the kidney itself, within each kidney. So here it is represented in a more simplistic fashion. So you can see here from the beginning till the end, you start off with the renal corpuscle, which each of these I'll get to in more detail just now. Then you go to the proximal convoluted tubule, and this is all situated in the cortex, or the cortical labyrinth. It will then go over to the spiral tubule, and you'll see this is in the outer stripe of the medulla. Then it will go to the thin limb of Henle. You can see it's thin. That makes a U-bend, usually in the inner part of the medulla. And then it will go to the thick ascending limb. You can see this part is thicker than that. It will proceed upwards, go out of the medullary ray. It passes the same renal corpuscle from whence it came, and then it becomes the distal convoluted tubule. At which point again it will enter the medullary ray, and it is now the collecting tubule. And at this stage is pretty much urine, which passes through here and out to the pelvis. Okay, so this is a lot of words, and there's no real easy way to remember this. You just have to sit down and you have to make a mind map, or you have to visualize it or something, and you've got to remember the order of these structures. Okay, and that is far easier to do if you have a sketch to put it onto. So for example, this particular sketch I could give to you and ask you to label the particular parts. Okay, it makes a lot more sense than saying just list the order of these tubules. Okay, but you really need to get this information into your heads, because once you've got the order, where they are and where they belong, it'll be a lot easier to remember the information attached to each particular part. So I just want to have a look at the first one, and then we'll take a short break. So the first one we're looking at is the renal corpuscle. So here it's written as Bowman's capsule, but the renal corpuscle is this entire structure here. Okay, a corpuscle is normally a round structure, and you'll see that these structures are quite round. This green part over here is the Bowman's capsule, and this part is the glomerulus. Now remember, the kidney's one function was to regulate the composition of the blood. So how does it actually do that? So there's fluid, and there's toxins, and there's other products which need to be excreted that are in the blood system, in the vascular system, dissolved in the fluid. So how does that get into these tubules I've been talking about? So obviously at some stage the blood has to be taken to those tubules, and this is where it all starts. So here you can see is a blood vessel going in. It then forms a whole network, which is called a glomerulus. So essentially, you have one arteriole coming in, it then branches, and the blood flows through there, and where it comes out the other side, it is single again. Okay, so the one that goes in is called the afferent arteriole, and the one that goes out is called the efferent. And this happens for each tubule. So if an animal has 500,000 tubules in the kidney, there will be 500,000 of these structures in that one kidney. So if you have a look at this is a three-dimensional structure, and that is called Bowman's capsule. Now can you imagine a balloon which is blown up? And then you take your fist, and now the balloon is not too blown up too hard, and you take your fist and you push it into the balloon. It's going to indent that balloon. So if your fist was the glomerulus and you push it into that balloon, then the balloon represents the Bowman's capsule. So now you have a part of that balloon surrounding your fist, and you have a part of the balloon which is on the outside, and there's air in between. So that part of the balloon which is around your fist, so essentially that part of Bowman's capsule which is around the glomerulus, which is all the blood vessels, is called the visceral epithelium. That part of the balloon which is on the outside and doesn't contact your fist, that's called the parietal epithelium. And you can see that on the outside. It's all one structure and it's continuous with itself. It's not two different things. So here you have parietal epithelium. It consists of a simple squamous epithelium. So simple, so there's only one layer of cells, and squamous being flat. So that's what I've tried to show here. So these are nice flat cells. The visceral epithelium is very specialized, and that epithelium is tightly associated with the blood vessels. So if you could think of a sieve, you know, like you get a sieve for flour, and you use sieve flour, and you were to squash an apple through there, what comes out the other side would be apple puree. So the glomerulus is almost like that sort of structure. So you have your blood coming through, but you actually don't want to squash out the blood cells, otherwise you have a problem. So you want the fluid and the other products to come out, but you do not want the blood cells to go through there. So this is almost like a sieve. So the holes that are present are only small enough for small molecules to get out, and not big enough for blood cells to get out. Obviously, if the animal experienced trauma and this particular structure was damaged, it would be possible that blood cells could get in. Okay, but if you see blood in the urine, it could come from a number of places, and the more likely place it would come would be from the bladder. If it comes from the kidneys, there's serious issues with the dog. So this entire structure here is the renal corpuscle, consisting of Bowman's capsule with its visceral epithelium around the blood vessels and the parietal epithelium on the outside. It then continues as the proximal convoluted tubule. And that tubule is lined by a simple cuboidal epithelium. Okay, so you'll see between there and there you have a flat cell and then suddenly a square cell. Okay, so here it is just in another picture. Shows it quite nicely. You've got your blood vessel coming in. So there you have your afferent arteriole. It then branches and becomes this whole convoluted structure you see here. So there's a number of little blood vessels which form the glomerulus. And here you can see Bowman's capsule, the parietal part, which has got flat cells. And then here you can see the visceral epithelium, which is closely associated with these blood vessels. So what will happen is the part that goes through that filtration membrane, which is the glomerulus and the visceral epithelium of Bowman's capsule, is called the filtrate. And that enters into this space. So essentially where you've got the air in between those two layers of the balloon, you can think of that, which is the space into which the filtrate goes. Now if you were to stick a hole or a pipe in there, that would then be the proximal convoluted tubule. So here you can see these nice cuboidal cells at the beginning of the proximal convoluted tubule. What are those three words telling you? It's proximal. Proximal means closer towards the origin. Sorry. It is convoluted. Okay, you don't see that here, but essentially it's going to be twisting and turning. You'll see that just now. And then it's a tubule. Okay, so obviously it's a nice hollow tube. It's a nice hollow structure. So that is where the filtrate will be moving down. So that's one way in which you can try and remember the names of these tubules and the order in which they occur. The proximal one will obviously be first. Okay, so I think at that point, okay, this is just what it looks like under the microscope. Again, you won't be asked to recognize this, but here's the sketch. There you see the renal corpuscle, and that's what it looks like under the microscope. Okay, so you can see it's a nice round structure. There's a parietal epithelium, visceral epithelium, and the glomerulus, or the capillaries, and there it goes into the proximal convoluted tubule. This is just for interest. Very pretty picture. This is a scanning electron micrograph. So these podocytes is another name for the visceral epithelium of Bowman's capsule. So these are the epithelial cells which are tightly associated with the glomerulus. So those capillaries. So if you have a look at these little slits over here, or these little interdigitations, like if you put your fingers together, these are the filtration slits through which the fluid will move. So blood vessels cannot get through here. Protein molecules are also too large to get through here. But you do get conditions when animals are ill where they end up with protein in the urine. Okay, there shouldn't be too much protein in the urine, but if the structure is damaged, protein can get in. But these are very fine little slits, so you can think of that like the holes that you find in the sieve. Okay, so with that we'll take a break. You can... Okay, so we've looked at the beginning of the nephron, which is the Bowman's capsule, which is just this part over here. The renal corpuscles, this entire structure with the blood vessels. So now we're going to look at the proximal convoluted tubule, and that part which is the spiral tubule. Okay, a nice busy slide. Again, all these pictures are just here to show you what the words say. Okay, so the proximal convoluted tubule has a cuboidal epithelium, whereas the thin or descending limb of Henle has a squamous epithelium. Okay, so that's why it's also called the thin limb, because the actual size of the tube is narrower. It's called the descending limb, so it's either the thin or the descending, because it's going down. It's going from the cortex, which is on the outside, and it's moving down towards the medulla. So that entire structure is a U-shaped structure. So the whole U bend is called the limb of Henle. So there, the proximal convoluted tubule. The spiral tubule is that part in the medullary ray and the outer stripe. So the transition of the spiral tubule to the thin limb or the descending limb of Henle then gives that the definition that you see between the outer stripe and the inner stripe of the medulla. Okay, so remember your medulla, the first part is the outer stripe. So what are you seeing here? Spiral tubules mainly. So there's a spiral tubule, there's a spiral tubule. Remember, it's lined by a cuboidal epithelium, and you can just make that out over here. There's a nucleus, there's a nucleus, there's a nucleus, and these cells have a nice square cuboidal shape to them. Okay, so here we have it. In the outer stripe, you have the spiral tubules with the nice cuboidal epithelium. Whereas on the inner stripe, you have the thin or the descending limb of Henle, and that has a flatter epithelium. And that gives this effect over here, where you can see an obvious difference between the outer stripe and the inner stripe. So you need to know this in theory, but remember, you don't have to be able to identify this. But this little sketch over here, you should know. Okay, so I've already mentioned to you the limb of Henle. So it's this nice U-shaped structure. Now this can differ in length in different animals. Now if you think of an animal that lives in the desert, does it have regular access to water? Okay, so it's not getting a lot of water. So how does it then regulate its blood composition? How does it not dehydrate? So one of the things that it does is it has a very long loop of Henle. So normally the longer the loop, the more it can concentrate its urine. So essentially more concentrated urine would mean it has more excretory products but less water. So the animal is much better able to concentrate its urine. So therefore, because it's passing less water, it has to drink less. So it loses water through evaporation through its skin, and it will lose water through the urine. So if it can limit the amount of water it can lose through the urine and limit the amount of water that evaporates from its skin, it will then be an animal that's adapted to living in the desert. So it doesn't have to take as much water in.
But can bladder stones form more easily then?
Pardon?
Can bladder stones form easily then?
In more concentrated urine. Sure.
Those type of animals is more common.
I don't think so. The kidney stones or the bladder stones are normally things that you see in domestic animals. I don't think they form so readily in your wild animals. The domestic animals that you have are generally not desert animals, other than I think it's the chinchillas, which can concentrate their urine quite a lot. But I mean, I suppose technically they should, but it also bladder stones or kidney stones would also depend on the pH of the urine. So often if the animal's diseased and the pH changes, it can then cause those to precipitate out. Okay, so those animals then which have free access to water, or that live in water, for example, a beaver, which lives in water, will have very short loops of Henle. So its ability to concentrate the urine would be limited, but it's really not necessary, because a beaver has to live by water. Okay, so it's got free access to it. So the loop of Henle is pretty much what determines how concentrated the urine can become. So here you have the spiral tubule, which at the junction between the outer and the inner stripe becomes the thin or descending limb of Henle. It will then turn around depending on how long it is, normally in the inner zone and the medulla. And then between the inner and the outer zone, you'll have its transition to the thick limb. Okay, so why is one called thin and one's called thick? Because it's exactly what they are. So this one is thinner and it's got the squamous epithelium. This one has got a lot thicker lumen, and it also has the thicker epithelium, and it goes back to the cuboidal epithelium that we saw in the spiral tubule. Okay, so there you can see this tubule is going up, and it goes out of the medulla into the medullary ray. Okay, so there you can see it going into the medullary ray, and then it will enter into the cortical labyrinth. So here we have what it looks like in the inner zone. It's a histological picture, so it's this little part down here. So what would you expect to find? Okay, you would find the loops of Henle. So you would find the thin or the descending limb, and then you will also find the part that's going up, but you're not going to see whether it's going down or it's going up. So you will only see the thin limb. And there you see it, and sometimes, which it doesn't show here, you will even see the U-shaped bend. So this one you can see has got nice flat epithelium, so that's a thin limb of Henle. This is a blood vessel, because remember there's obviously got to be blood vessels in this tissue. Looks very similar to the thin limb, but you don't need to know the difference. And then here you have a collecting duct, which is the final portion, which we'll get to just now. But you can see the collecting duct has an obviously different epithelium to that of the thin limb of Henle. So in this part of the inner zone, the only thing you will find are the thin limbs of Henle, so either the thin ascending limb or the descending one, and you will find collecting ducts. Okay, this is just it in cross-section. So here you can see quite nicely these thin limbs and the collecting ducts with their nice cuboidal epithelium. There's the epithelium of the thin limb, and you can see that all over here. Okay, now remember, just before it goes into the medullary ray, what are you going to find in the outer zone of the medulla? Okay, so what was going down there? You had the spiral tubules before it became the limb of Henle. And then the limb of Henle, as it goes up again, becomes the thick ascending limb. So that's exactly what you're finding here. Here you have your spiral tubules, and here you have your thick ascending limb of Henle. Okay, so don't stress about having to recognize it. I just want to show that what I'm telling you is not entirely rubbish, although you would have to take my word for it that this is the thick ascending limb. But you can tell that by looking at the epithelium. So here's a cuboidal epithelium, but it's a lot lower than this epithelium. So we've done the Bowman's capsule, the proximal convoluted tubule, it's the first one, it's all convoluted, the spiral tubule as it goes down to the medulla, then the nice U-shaped thing, the limb of Henle, its thin limb and the thick ascending limb. Now we're looking at the distal convoluted tubule. So what's that going to tell you? Again, that it's convoluted, so it's all spiral. So that tubule you're going to find in the cortical labyrinth, because remember, what was a labyrinth? The labyrinth was that part which is all spiraled and forms like a maze. And it's distal. So obviously this one is going to be closer to the end point than the proximal one. So the way I've ordered this slide, unfortunately, it goes from the bottom to the top. You can see 1, 2, 3, 4. So the ascending limb goes up the medullary ray. Then it goes towards the renal corpuscle from which it originated. And that's what you can see here. So it goes on this whole journey throughout the kidney, and it ends back right at this point. And at this point, it forms a specialized structure called the macula densa. It's essentially like a bunch of sensory cells. So this region here, opposite the renal corpuscle, is called the macula densa. And then at a particular point past that, and there's no specific point at which it occurs, you actually have to look at it histologically, it then becomes the distal convoluted tubule. So there you can see the convolutions, and this is the distal one. And that is the final part before it goes into the collecting tubules. And the collecting tubules is essentially what's collecting the final product, and that would be the urine. And that then goes again into the medullary ray. So in the labyrinth, what do you find? The renal corpuscles and your convoluted tubules. That's essentially what you're going to be seeing in the cortical labyrinth. Now we just have to look at this from a little bit of a distance again. But there's your entire nephron. And remember, once it goes to the collecting tubules, then it includes the renal. Now I've just lost the term. The renal tubule. Okay, so the renal tubule would be everything that you see in here, whereas the nephron is only that portion from Bowman's capsule up until the collecting ducts. So you're going to have hundreds of thousands of these structures and tubules, and they're all going to go into the collecting tubules, which passes down the medullary ray and will pass down the entire length of the medulla. So you will find these collecting ducts all throughout the medulla. And they finally open up onto the larger ducts of Bellini, which then open up into the renal pelvis. Yes?
The limb of Henle on the right-hand side, would that be of an animal that's more with water about?
Yes, so that would be an example of a short looped limb of Henle, and this would be an example of a long looped one. So the longer the loop, the more it's able to concentrate the urine. Okay, so the blood supply. So the blood supply obviously comes in the renal arteries. Okay, in dogs, about 25% of dogs have a double renal artery. So there's not just one renal artery going to the kidney, there can actually be two. Okay, and these arteries come directly off the aorta. Okay, I think you should all know what the aorta is. That's the main blood vessel that comes from the heart, and it runs down the dorsal midline of the animal. And so these renal arteries are pretty short because the aorta runs down the dorsal midline and the kidneys are situated either side of it. So the arterial blood will enter through here. Obviously, there will be renal veins through which the blood will drain. So that will enter the kidney, and at a point, it will form these arteries here, which you don't have to know the name of, but basically at the junction between the medulla and the cortex, you have these arteries, and they give off the interlobular arteries, which you see here. So between each of these medullary rays, you have an interlobular artery. So inter is in between these lobules. And coming off there, you have your afferent arterioles. And each afferent arteriole will then go and form the glomerulus, which is associated with Bowman's capsule, and then it will then exit as the efferent arteriole. Okay, so that's what you can see here. And then what's demonstrated very clearly is this interlobular artery then also supplies the capsule. It'll end up at the end of the cortex and it'll supply blood to the capsule. Someone asked earlier about those blood vessels, or on the cat's kidney, whether it was lobulated or not, and you saw those blood vessels so clearly. So those would be part of this arterial system. Okay, so that's all you have to know about the blood supply of the kidney. But the most important one is that the afferent arteriole will supply the glomerulus, and the efferent arteriole is the one which exits. Okay, here you can see afferent going in, efferent going out. Remember I mentioned about the macula densa. So this is the part of the thick ascending limb of Henle, just before it goes over to the distal convoluted tubule. So this structure, together with these cells around here, forms what's called the juxtaglomerular apparatus. Okay, juxta means next to. So here you have this apparatus which is next to the glomerulus, associated with it, and it is responsible for monitoring the blood pressure and then also secreting renin, which then regulates the blood pressure. Okay, so here you can see specifically renin will raise the blood pressure. So if the blood pressure is raised, what will that do? That's going to put more force behind the blood flowing in the glomerulus. So that should, in theory, squeeze out more fluid. So if you think if you're squashing cooked apple through a sieve, the harder you press, the more you're going to get out. Okay, so renin will increase the blood pressure, and it will cause more filtrate, more urine. Okay, so renin is obviously going to be released when the blood pressure is too low. Right, then the last part, the last slide, which I'm sure will make you all very excited. Transitional epithelium. Have you heard of that before? Yes, Prof. Soli should have gone through it with you. So this type of epithelium which lines the urinary system, so that would be the renal pelvis, the ureters, the bladder, and you also find it in portions of the urethra, is called transitional epithelium. So that specifically is a pseudostratified. So pseudo means fake, it's false. Stratified means many layers. So it looks like it has many layers, but in actual fact, each one of these cells contacts the basement membrane. So it is a pseudostratified epithelium, specifically called transitional, and it is designed that it can stretch. So obviously, the bladder is going to be full or empty, depending whether it has urine or not. So how does it do that? It's stretchy, it's like a balloon. Okay, so this particular epithelium gives it properties almost like you have for a rubber balloon. It has the ability to stretch. So this is what the epithelium looks like in the relaxed state. And when it is stretched, all those cells flatten out. And that allows the organ, which is the bladder, or even the pelvis and the ureters, to stretch and accommodate an increased amount of fluids. So, what are you looking at for the pelvis, ureter, and bladder? You have a transitional epithelium. I've just been talking about you. They say they remember what you told them. Almost done. Okay.
Taking breaks soon?
No, almost done.
So shall I just go back?
Okay, so we have the transitional epithelium supported by lamina propria, which would just essentially be connective tissue. And then you have a tunica muscularis. Okay, so that's the muscle layer, which is obviously going to give the ability of the structure to contract. And then just the supporting connective tissue outside called the adventitia. So again, this would be something that would be requested of you to know, are the layers of these structures. Okay, so you might be asked to name the layers of the ureter from within outwards, and that would then be the transitional epithelium, lamina propria, tunica muscularis, and adventitia. But try and add some sort of meaning and sense onto these words, and try and learn it with the pictures, because it'll make it a lot easier to recall. Okay, so that is pretty much my story with the kidneys. I'm sure that was perfectly scintillating. You all look more awake now than when I started, but I think it's because I said I'm almost done. Okay. All right, so I'm going to end. If you guys have any questions, you're welcome to ask me